Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the uptake of molecules or the movement of molecules within the cell, right? So far what we have discussed about the different processes which are required for the transport of material into the cell. In this context, in the previous two lectures we focused mainly on the transport, what is happening from outside the cell, right? So, if you see the transport, right, the transport is very essential for the many type of functions. Like transport is essential for taking of the nutrition. Transport is important for the driving the cell signaling and so on. So far, we focused only on to the uptake of material from outside, right? And in that process, we discuss about the diffusions, we have discussed about the osmosis, we discussed about the active transport, we have discussed about the passive transport, and so on. Now, in today's lecture, we are going to focus on to the transport of material inside the cell, right? And within the cell also, right? Because you are not going to have the transport only from outside, but you're also going to have a transport within the cell and within the different organelles as well. So when you talk about a cell, right, it actually requires to uptake the molecule which is from outside, right? So when it crossing the outside, right, it is actually crossing this particular barrier, right? And depending upon the chemical property or the structural property of this molecule and the chemical property of the barrier, there are different methods what the molecule is going to adopt or the organism is going to adopt so that this molecule will get inside the cytosol. Now, once it get into the cytosol, then it has to be distributed to the different parts of the cell, right? Then it has to be distributed to the its final destination, right? Its final destination could be mitochondria, final destination could be lysosomes, final destination could be nucleus, and so on, right? And it could be a final destination that it has to be in the cytosol. So in today's lecture, we are going to discuss about the intracellular distribution of the molecule. So how the transport is occurring inside the cell, how this molecule is transporting to the mitochondria, how this molecule is transporting to the nucleus, and so on. So in the transport of the material within the cell, it's actually being considered as the, so when the molecule is going to be internalized, it is going to be utilized for these kind of intracellular transports. So endocytosis is a process by which the eukaryotic cell internalize the molecules such as the plasma membrane components, fluids, solutes, macromolecules, and the particulate substances. Example is if you are suppose taking the glucose, if you are taking the particulate matter like bacteria and so on. The internalized cargo may include receptor ligand complexes, various nutrients and their transporters, component of the extracellular matrix, cellular debris, microbes such as bacteria and viruses, and in specialized cases, even the whole cell. For example, if the one cell is getting apoptosed and other cell is going to take up the whole cell so that it can actually be able to digest that particular cell and then it redistributes the nutrients what is present. This process also allows the cells to regulate the composition of its plasma membrane in response to the changing external conditions. Endocytosis can generally be divided into two broad categories, phagocytosis and the pinocytosis. Phagocytosis means eating, and pinocytosis means drinking. So further, various different classification of pinocytosis has been proposed, and the classification of endocytosis is that it is broadly being classified into two processes. One is called phagocytosis, the other one is called as the pinocytosis. Phagocytosis means eating the particles, right? And this phagocytosis we are going to discuss in detail when we are going to discuss about the immune responses. Then we have the pinocytosis. Pinocytosis means drinking actually, or drinking the fluids. Now, pinocytosis could be of two types. It could be a Clathrin-dependent process or Clathrin-independent process, right? So, Clathrin is a protein, right? And within the Clathrin-dependent process, you can have the Clathrin-mediated endocytosis, right? And then within the Clathrin-independent process, you can have the Caveola-mediated processes, you can have the Caveola and Clathrin-independent process, and then you have the Macropinocytosis. And within the Caveola and Clathrin independent processes, you can have the ARF associated endocytosis, you can have the Clicks or Geeks pathway, you can have the Floetin associated endocytosis, or you can have the row dependent endocytosis. Now, first talk about the endocytosis. So, endocytosis is the transport of the molecule within the cell, right? So, the cargo that is being transported is enclosed by a small portion of the plasma membrane by invagination and then pinching off to form an endocytic vesicle that contains the cargo. So, when the molecule will enter, it is actually going to form a vesicle, right? It has to be trapped within the vesicle, and this vesicle is being called as the endosome, right? Most of the endocytic vesicles and then fuse near the periphery of the cell with the receiving compartment termed the early endosome. It is the primary sorting station as the internalized cargo is sorted. The endosomal system is complex and consists of many tubular and vesicular membrane-bound compartments. The tubular region of the early endosomes form the vesicles that return internalized cargo to the plasma membrane directly or by a recycling endosome. Rest of the cargo is marked for degradation and directed to form the late endosomes. Endosome maturation is the process by which the late endosomes are formed from the vacuolar portion of the endosome. Thus, as early endosome mature into the late endosome, their tubular extensions disappear. So, this is what it is showing here, right? You are actually going to have the internalization, and that's why you're going to form the early endosomes. And then this early endosome will go through a definite steps of processes, and that's how it is actually going to form the late endosomes, and then late endosomes are actually going to fused with the lysosomes to form the endosome lysosomes, right? And because the lysosomes has the degrading enzymes, right? It has a digestive enzyme, it has a low pH, it is actually going to degrade all the material what is being taken up from the outside, and that's how this material is now be ready for redistribution within the cytosol or the other organelles. So the maturing late endosomes, also known as the multivascular body, is transported along with the microtubule towards the cellular interiors. And when the endosome mature, it stops recycling material to the membrane, and all the remaining content is designated for the degradations. Late endosomes fuse with the existing lysosomes to form the endolysosomes. Lysosomes are the membrane-bound organelle containing the hydrolytic enzyme that break down the macromolecules. The degradation of the cargo takes place in the endolysosome, and endolysosomes mature into lysosome hydrolysis, finishing digesting their content including the intracellular vesicles. Now, all the stages of the endosome maturations are linked via the bidirectional vesicular transport process to the trans-Golgi complexes. They are connected by the transport vesicles with the trans-Golgi network, and the trans-Golgi network sorts the protein based on their final destinations. These are actually the part of intracellular tracking, right, and which we are not discussing in detail about this, right, because very briefly we discussed when we were discussing about the cellular structures, and now also we are not getting into the molecular details of these kind of processes. But if you are interested, there are so many excellent courses about the cell biology, where the instructors are only focused on to only these aspects. So there are so many courses available on the hardcore cell biology courses, so that can be followed. These pathways deliver the newly made lysosome enzyme from the ER back to the early secretory pathways. Now, from the endocytosis, it could be divided into phagocytosis or the pinocytosis. Let's talk about the phagocytosis very briefly, because in detail phagocytosis we are going to discuss when we are going to take up the immune system. So phagocytosis is the uptake of large particle like bacteria. It is also called as the cell eating. It occurs primarily in a specialized cell type. So you know that these are like going to happen in the all sorts of antigen presenting cells like macrophages, dendritic cells, B cells, and it is a type of endocytosis where the cell utilizes the large endocytic vesicles termed as the phagosomes. So phagocytosis is a process through which you are actually engulfing the material which are very large in size from outside. Mostly the phagocytosis is being used for eliciting the immune responses or protecting the cell from the infectious organisms like bacteria, fungi, viruses and so on, and they are being used for eliciting the immune responses, and that's why the vesicle which are going to be formed after the phagocytosis is known as the phagosomes. They are used to digest the particles like microorganisms and dead bodies. It play a role in ingesting the alimenting particle which are larger than 0.5 micrometer, and so if the molecules are bigger than this, then it is going to be called as phagocytosis. So upon binding of the particle to the receptor on the surface of the phagocytosis, so basically the phagocytosis could be receptor mediated phagocytosis or non-receptor mediated phagocytosis. Mostly the receptor mediated phagocytosis is more efficient, whereas the non-receptor mediated phagocytosis is less efficient, just like as we discussed about the the facilitated diffusion versus the normal diffusion, right? Even the passive, both are passive, but facilitated diffusion is much more efficient compared to the normal diffusion. So extension of the pseudopodia is induced, which in then actin based movement of the cell surface. Pseudopodia surrounds the particle and then the membrane and then fuse to form a phagolysosome. So basically what happen is that you have a particle, right? And so what happen is the cell will actually going to approach first. If it is a receptor mediated phagocytosis, it is actually going to bind the particles with the help of the receptors. So there will be a ligand what is present onto the this particular object. For example, if there are bacteria, then it may have the bacterial associated antigens, and these antigens are going to be recognized by the receptor what is present onto the cell surface. It could be the toll-like receptors, it could be Fc receptor gamma, or and it could be IL receptors, it could be interferon gamma receptors and so on. So once it recognizes, then it is actually going to start spreading its pseudopodia, right? It will start spreading the pseudopodia across this, right? So it is going to start spreading the pseudopodia from both sides, right? And ultimately these pseudopodia which are coming from the cell are actually going to be fused, and then this particle will come inside, and that's how it is actually going to form a phagosome. So this is what exactly going to happen here, right? So the cell is actually spreading its particle, its pseudopodia around the particles, right? And then particle cannot move around because it has already been bound to the receptor what is present on the onto the cell surface, and then ultimately these pseudopodia are going to be fused, and that's how it is actually going to be present into a bilayer membrane, right? And that is being called as the phagosome. Now this phagosome will go through a process of maturation, just like as we discussed for the endosomes, and then it is actually going to fuse with lysosomes, and then it is going to form the phagolysosomes, and these phagolysosomes are then going to be used for digesting the material, right? So it is going to digest the bacteria, and it is going to digest the bacteria into small pieces, and these small pieces are the peptide fragments are then going to be expressed along with the MSC, and that's how it is actually going to give the signal to the body that how the if there is an infectious organism, we should actually now start sending the immune responses. So phagosomes fuse with lysosomes to form the phagolysosomes, where the ingested particles are digested by the hydrolytic enzymes what is present in the lysosomes, and the phagosome maturation then recycles some of the membrane protein back to the plasma membrane. So many amoeba use the phagocytosis as a feeding mechanism by the capture of food particles such as bacteria or the protozoans. Large particles engulfed into the phagosomes are delivered to the lysosomes, where they are digested and resulting into breakdown products enter the cytosol for use as a nutrient. So Phagocytosis is a generalized mechanism through which you are actually uptaking the large particles from the surrounding. Now let's move on to the next process, and the next process is the pinocytosis. Pinocytosis means drinking, right? So remember that drinking is always been done for the fluid, right? Not for the particulate matter. If you are doing the particulate matter, then it is going to be for phagocytosis. This means the size of these particles has to be less than 0.5 micrometer. Pinocytosis. The pinocytosis is the uptake of fluid or macromolecule in a small molecule. It is also called as cell drinking. It can occur in all eukaryotic cells and include the various different mechanisms. Nearly all eukaryotic cells constantly take in small part of their plasma membrane through pinocytosis or endocytic vesicles, a process in which the extracellular fluid or small particles are enclosed in a membrane-bound vesicle and internalized into the cell. So basically, you have the extracellular fluid, right? This fluid could be containing the very small particles like glucose, fructose, all those kind of molecules. And then what happens is the cell is going to, get into invaginated, right, inside, and then it is going to be trapped, right? And because of this, this particular vesicle is going to be formed, and this vesicle has actually taken up the material what is present here, right? What is present in the environment, right? So you see this green color particle, right, been taken up. So this green color particle is going to be taken up, and then it is actually going to form the endosomes, or it is going to be called as endocytic vesicles. And then these vesicles are then again go through the process of maturation. So pinocytosis could be of different types. It could be a Clathrin-mediated endocytosis, it could be Caveolae-mediated endocytosis, it could be Clathrin or Caveola-independent endocytosis, or the micropinocytosis. So this is the micropinocytosis, this is the Clathrin-mediated pinocytosis, this is Caveola-mediated, and this is the Clathrin and Caveola-independent processes, right? So in this case, you are actually going to use the Clathrin proteins. Here you are going to use the caveolas, and here you are not using anything. So first talk about the Clathrin-dependent endocytosis. So it is involved in selective uptake of specific macromolecules. It is involved in cargo transport in virtually all setup, including the turnover of the plasma membrane components, internalizing the nutrients like the LDL, and the iron saturated transferrin, and the endocytosis of many growth factor receptor after they activated. Pathogens like influenza viruses and the bacterial toxins such as Shigella toxins exploit the Clathrin-mediated endocytosis to enter the cells. So basically, Clathrin-mediated endocytosis or the pinocytosis is being used for uptaking the small molecules, uptaking the growth factors, viruses, and the toxins, bacterial toxins, right? So that all these are part of the fluids. All these part are considered to be fluid, right? So for example, the viruses also. Viruses are very small particles, so they are not going to be taken up by the phagocytosis. They are going to be taken up by the pinocytosis. So the cargo binds to the specific cell surface receptor. Most of these cargoes are present in a specialized region of the plasma membrane termed the Clathrin-coated pits. Dynamine, a protein associated GTP binding protein, helps the pit bud from the membrane to form the small Clathrin-coated vesicles, which contain the receptor with the bound ligand, that is the cargo. The Clathrin-coated vesicles fuse with the early endosomes, which leads to sorting their content for either recycling to the plasma membrane or transport to the lysosomes. Clathrin-mediated endocytosis selectively capture the ligands using the receptor. This increases their internalization efficiency of ligand more than 100-fold, leading to the uptake in large amount of even minor component of the extracellular fluid. An example of Clathrin-mediated endocytosis is the uptake of cholesterol in mammalian cells. Through this endocytosis, they can acquire the cholesterol to make the new membrane. Okay, and in this process, you are actually going to use the LDL receptors and all that. So transport of most cholesterol in blood is a cholesterol ester in the form of lipid protein particles known as a low density lipoprotein, or the LDL, right? You remember that we have the three different types of fats, right? High density lipoproteins, low density lipoprotein, and very low density lipoprotein. So we have the LDL, we have the HDL, and we also have the VLDL, right? The diagrams on right side show the one of the LDL particles, right? So core has approximately 1500 cholesterol molecules. So within this, you see this is the cholesterol molecules that are esterified to the long chain fatty acid. So these are the cholesterol molecules. These are the cholesterol molecules. The core of the cholesterol ester is surrounded by a lipid monolayer consisting of 800 phospholipid and 500 cholesterol molecules that are not esterified. You have the lipid bilayer, which contains the phospholipids, right? And then you also have the cholesterol molecules which are present in this particular membrane. And then you will also have a protein part which is called as the apolipoprotein B, play an important role in the organization of the particle and binding of the LDL to the LDL receptor onto the cell surface. And this core is being, so all these lipid particles are organized around the protein part, which is called as apolipoprotein B, right? And that's how this is the kind of a complex particle which are going to be formed, and all these particles are being classified as the LDL particles or the HDL particles or the VLDL particles, depending upon the composition, depending upon the type of cholesterol present in this. So what is the structure of the LDL receptor? So you see this is the structure of the LDL receptor, where you have on the N terminus you have the LDL binding domain. So this is the LDL binding domain, which is actually extracellular, right? And then we have the N-linked glycosylation. And then we have the O-linked glycosylation. All these we are not discussing, but N-linked glycosylation, where the N is going to be utilized for linking the sugar molecules, whereas O-linked glycosylation, where the oxygen present onto the amino acids are going to be utilized for attaching the glucose molecules. Then we have the plasma membrane. So this is the bilayer, right? And this is the transmembrane region. And then you also have the cytoplasmic tail. Cytoplasmic tail is going to be used for driving the downstream signaling so that it can be, uptake. So as soon as the LDL particle will come and bind to this, right, it is going to be this whole LDL and the LDL receptor complex is going to be internalized by the process of endocytosis, and then it is actually going to be this deliver the LDL into the cytosol, and then this receptor is going to be recycled and it will reappear onto the surface. So it consists of 700 extracellular amino acid, a transmembrane alpha helix containing of 22 amino acid, and a cytoplasmic domain with the 50 amino acid. So see, the large number of, or more than 90% protein is present outside. Very small fraction is present inside. The N terminal form the LDL binding domain. So this is the LDL binding domain. Internalization of signal is present in the cytoplasmic tail. So this actually is a cytoplasmic tail which is actually going to bind the scaffolding proteins and downstream machinery, and that's how it is actually going to pull the LDL and the LDL receptor complex into the vesicles. So the cell synthesize the LDL receptor when it requires the cholesterol, right? And these receptor proteins are transmembrane, and when they are inserted in the plasma membrane, they diffuse until they are associated with the Clathrin-mediated pitch, which are in the process of forming. So what happens is that you are actually going to have the LDL receptor, right? And then LDL receptor is going to be coated with the Clathrin molecule. So you are going to have the aggregation of the Clathrin molecules around this particular pit where the LDL receptors are coming together, right? And you see the other side, the LDL is already bound, right? So this is the LDL molecule which is bound. And then we are actually going to have the Clathrin coated vesicles. So these vesicles, which are actually these Clathrin individual molecules, they are coming and coating this particular part, and that's how they are actually going to make a vesicle. Now this vesicle is going to be pulled onto the downward side. So when it actually going to pull, it is actually not going to pull only the vesicles or the protein. It is also going to remove this portion also, right? So it is, this whole thing is going to be internalized, and that's how it is actually going to form the vesicles, where the LDL receptor and the LDL molecule is going to be facing downward, right? And then what happen is that it is all these are LDL particles are going to be delivered into the endosomes, whereas the receptor is going, because the Clathrin is going to be removed once this is going to happen, and then the LDL receptor is going to be recycled back to the plasma membrane, and that's how and it is actually going to deliver the LDL into the endosome. So upon receiving an endocytosis signal, its cytoplasmic tail, the receptor binds the membrane-bound adapter protein, which is called as AP2. Clathrin is then recruited by the AP2 to start the endocytosis process. Upon forming a Clathrin-coated vesicles, the LDL that is bound to LDL receptor interpret is internalized in these vesicles. The Clathrin coating are shed and the content of the vesicle is then sent to the early endosome. The low pH in the endosome causes the release of LDL from the receptor and it thus delivered to the lysosome through a late endosome. The cholesterol esters present in LDL are hydrolyzed to form the free cholesterol, which then can be used for the cell to synthesize the new membrane. So, once it is being present in the early endosome will mature and form the late endosomes. Late endosome is going to fuse with the lysosomes, and that's how the material is going to be recycled and cholesterol is going to be released into the cell, and then this cholesterol is going to be used as a building block to synthesize the new plasma membrane. If there is an excess of free cholesterol inside the cell stops its own cholesterol synthesis along with the LDL receptor synthesis. So basically, if the cell is synthesizing the large quantity of cholesterol, then it will actually going to down regulate the amount of LDL receptor what is present onto the cell surface, and that's how it is actually not going to remove the circulating cholesterol, right? So we have discussed about the Clathrin-mediated endocytosis. Now we'll talk about the Clathrin-independent process. So in the Clathrin-independent process, the first process is the Caveola-mediated process. So it's a Clathrin-independent process, but it is Caveola-mediated process. So in the Caveola-mediated endocytosis, in many cell types, the plasma membrane is covered by a small pit, which is called as caveola. They are small bulb-shaped plasma membrane imaginations, typically 70 to 100 nanometer in diameter, and that extend into the cytosol. They are present in many cell types such as adipocytes, endothelial cells, muscle cells, fibroblasts, and the astrocytes. The caveola coat includes a protein which play a role in formation and stabilization of the membrane invagination. Some of the protein includes the caveolin, which has three orthologous in humans, that is caveolin 1, 2 to 3. Then caveins, which include the cavein 1 to 4. And then we have the bar protein domain containing synartin or the plasticine 2. And then we have the dynamine radiated ATPases, which is called as EHD2. So this is the general structure of a caveola, right? Where you have the caveolin proteins, we have a caveins, we have the bar protein domain containing this, and then we have also dynamine related ATPases. Caveolins are oligomeric cholesterol binding integral membrane proteins. They can range in the size from 17 to 24 kDa and play an important role in the invagination of the caveolin from the plasma membrane. The caveolin protein family is encoded by the 3 genes. They consist of 6 caveolin subtypes like caveolin 1 alpha, caveolin 1 beta, caveolin 2 alpha, caveolin 2 beta, caveolin 2 gamma, and the caveolin 3. The structure of caveolin 3, it consists of 151 amino acids which forms the different domains, including the intracellular domains, caveolin scaffolding domains, oligomerization domains, and the signature motifs, right? So these are some of these things what is present, right? We have the intracellular domain, we have the caveolin scaffolding domain, then we have the oligomerization domain, and then we also have the signature motif. OD, that is the oligomerization domain, contains the CSM and the SM, is shown by a yellow box in the diagram. So this is what it is showing here, right? Whereas the intramembrane domain play a role in the insertion of caveolin oligomers into the membrane with the N and C terminal facing the cytoplasm. triggering the membrane structures and the mature caveolin formation along with the other proteins. So the caveolin play a role in membrane association, right? Cholesterol binding, oligomerization, and the regulation of signal transduction. The domain thus play a role in the interaction of the caveolin with the other signaling molecules. Caveolins are linked to each other by the oligomerization domain and form a disc-shaped oligomeric complex onto the membrane. The C-terminal can spirally assemble as the barrel-like channel. Some studies have suggested more domains in the protein, such as pin motifs, spoke regions, and the beta strands. These domains possibly play a role in forming the disc-shaped oligomeric complexes of the caveolins onto the plasma membrane required for the caveolin function. Caveolins play a role in lipid and fatty acid uptake, neuromuscular coupling, viral internalization, melanocyte pigmentation, and many other functions. Animal viruses like SV40 and the papillomavirus can enter the cell in vesicles derived from the caveolae. They are first transported to early endosome and then through the transport vesicles move to the ER lumen. The genome of the viruses then leaves the ER and goes to the cytosol and then transported to the nucleus where it can initiate the infection. Suggested mechanism for the caveolar internalization. So caveolae are dynamic membrane domain which can move literally into the plasma membrane. Both the EHD2 and fascism play a role in stabilizing the caveolin into the plasma membrane. Caveolin detachment from the plasma membrane is possibly caused by the EDH2 removal. It is possible that the dynamine might play a role here. After detachment, the caveolins are internalized and trafficked to the particular organelle. It is suggested that the various tissues and cell-specific signaling event or ligand may induce the internalization such as albumin, cholesterol, SV40, the virus, right? So this is all about the caveolin-mediated endocytosis. Now let's talk about the caveolin and Clathrin-independent processes. So Clathrin and caveolin-independent endocytosis. So endocytotic pathway that does not use the Clathrin and the associated molecular machinery are termed as the Clathrin-independent endocytic pathway or the CIES. The pathway can be constituted or induced by a specific signal or exploited by the pathogen. These pathways differ in the kinetics and mechanism of formation of endocytic vesicles, the molecules involved, and the destination of the transported cargo. Now, within this Clathrin-independent endocytosis, you can have the dynamine-independent process or you can have the dynamine-dependent process. So, dynamine is a row-dependent proteins, right? So, within the dynamine-independent process, you can have the ARF6 associated endocytosis, you can have the GECC pathway or you can have the flotlin associated endocytosis. Now, as long as the ARF6 associated endocytosis, so ADP ribosilane factor 6 or ARF6 is a GTP binding protein. It is present onto the cell surface in mammalian cells. It is also present on the endosome that have the cargo protein from the Clathrin-independent pathway. Proteins that utilize this pathway include GLUT1, CD98, LAT1, CD44, and CD147. Although the internalization is independent of Clathrin and dynamine, it is dependent on to the cholesterol. Then what is the mechanism? So ARF6 has two states, the inactive GDP bound state and the active GTP binding state. Active state in the plasma membrane helps in the internalization of cargo by utilizing the energy from the GTP hydrolysis mediated by the PIP2. Inactive state then associated with the tubular recycling endosome. Guanine nucleotide exchange factor like the GRP1 associated scaffolding protein can cause the activation of the ARF6. So basically in the basal inactive state, it has always been having the GDP binding state, right? ARF6 GTP then return to the plasma membrane and leads to the formation of protruding actin-rich structures which helps in recycling membrane back to the cell surface. As this endocytic route requires the cholesterol, the ARF6 associated endosomes are rich in cholesterol and PIP2. It is suggested that ARF6 activation is not required for endocytosis but play a role in recycling. The endocytosis has a role in regulation of the cell adhesions, migrations, tumor invasions, and the cytokinesis. Any DD9 is a marker as well as the target for the metastatic breast cancer. It inactivates ARF6 and thus inhibits the recycling of MMP14 to the plasma membrane. This increases MMP14 activity and facilitates the tumor invasion. These all things we are not discussing how the MMP14 is helping the tumor invasion and all that. And then we have the GECC pathway. So in the GECC pathway, we have the glycosyl phosphoinositol anchored protein used as a dynamine independent pathway for the endocytosis. They are endocytosed in a specialized early endosomal compartment. The endocytic structures which contain the endocyzed GPI anchored proteins are called as the GECC or the GPI anchored enriched early endosome compartment. They are formed by the fusion of primary uncoated Clathrin-independent tubular vesicle carrier called CLIC, which are derived from the surface of the cell. The pathway is then termed as the GLIC-GECC pathway. Suggested mechanisms: a guanine nucleotide exchange factor or GEF for ARF1, GBP1 is brought to the cell surface. It activates the ARF1, which then recruits a Rho JTPase, activating the protein to the cell surface. The complex play a role in maintaining the Rho JTPases CDC42 in its cycling state. CDC42 alternate between the active state and the inactive state at the plasma membrane, and this cycling is essential for the recruiting the actin polymerization machinery. GTPase regulator associated with the focal adhesion kinase 1 has the bar domain, SH3 domain, and a Rho JAP domain. It is another molecule possibly involved in the GEEC formation. The Rho gap domain can inactivate the CDC42 with the loss of the ARH gap 10/20 activity. The bar domain is possibly involved in generating the membrane curvature. It is suggested that after the CLIC are formed, they bud quickly from the plasma membrane, mature to the GEC stage, and then fuse with the early endosomes. Then we have the phlotellin associated endocytosis. So phlotellins are the protein that localize to the specific microdomain or the lipid rafts into the plasma membrane. There are two phlotellin genes, phlotellin 1 and phlotellin 2, which are possibly involved in inducing the membrane invagination in a dose-dependent manner. Phlotellin-mediated endocytosis is regulated by the SARC family kinases like FIN. Many studies suggest role of phlotellins in endosomal sorting process and formation of the exosomal vesicles into the endosome. Some studies also suggest their role in the cargo recycling. So, on receiving the stimulus, the dynamine phlotellin can oligomerize, which then recruit transmembrane protein like the EGFR, DAT, and the ARP. So, when you are unstimulated, when you are getting the stimulations, these proteins are actually going to be recruited onto the cell surface, and then they will form a pre-endocytic, clustering. So, they will actually going to form a clusters. Then the cargo is going to be internalized by the endocytosis without the involvement of protein, for example, by the Clathrin-mediated endocytosis. So then this whole thing is going to be invaginate with the help of the Clathrin-mediated endocytosis, and that's how this vesicle is going to form the endosome. Phlotellin is a cellular cargo sorting. So phlotellin microdomains are found not only onto the plasma membrane but also in early, late, and recycling endosomes as well as in exosomes. In the plasma membrane, phlotellins help the transmembrane cargo proteins during cluster formation before endocytosis, and described in the previous slide. So in endosomes, the protein possibly play a role in sorting the cargo for recycling to the plasma membrane, transport to the Golgi and ER or to the intracellular vesicles of the multivesicular body that later fuse the plasma membrane to release the vesicle as the exosomes. Then we have the Rho dependent IL-2 receptor endocytic routes. IL-2 receptors are concentrated and they internalize by small invagination that are not coated. Various cytokine receptor and their compartment like the beta chain of the IL-2 and IL-15 receptor and the common cytokine gamma chain of the receptor for internalizations of 2, 4, 7, 9, 15 and 21 utilize these endocytic routes. The endocytosis regulated by dynamin here is a for cholesterol sensitive. So, IL-2 receptors, endocytosis. The endocytosis is Clathrin and Caveolae-independent and dependent on the dynamin. So, this is the dynamin, right? And the pathway depends on the Rho small GTPases, Rho A, and it is regulated by the PI3 kinase and it's a lipid kinase. IL-2 activates PI3 kinase due to its internalization into a regulatory P85 subunits. This causes the production of PIP3 at the membrane. PIP3 then induces the VAV2, which is a guanine and nucleotide exchange factor for RAC. This causes the activation of the RAC. The RAC GTP then binds to the P85 and get recruited to the sites having the IL-2 receptor. The downstream target of the RAC1 P21 activated PAC1 is activated. So from this IL-2 receptor, what is present into the vesicles, then it is actually going to present into the early endosomes, then it forms the late endosomes and so on, and then it is actually going to fuse with the lysosomes. From lysosomes, it is going to be degraded, and that's how the material is going to be recycled. The cascade facilitating the recruitment and the activation of complex with the cortacine and VAP, ARP2/3 and leads to the generation of F-actin during the IL-2 receptor endocytosis. Upon internalization, they are suggested to be sorting in early and then to late endosome, lysosome and degraded or are recycled to the plasma membrane. So, we are already done with the Clathrin or the Caveolae-independent processes. Now we'll move on to the Macropinocytosis. Macropinocytosis is another process which is also Clathrin-independent process, right? So what is macropinocytosis? So macropinocytosis involves a non-specific uptake of large volume of extracellular fluid, solute and membrane into a large endocytic vesicle which are called as micropinosomes, right? So it is actually going to take up large amount of water or large amount of extracellular material. Large vesicles of the size of 0.15 to 5 micrometer in diameter are involved into the uptake process. So these are the micropinosomes which are going to be formed after taking up all these fluid which are extracellular fluid. It occurs in virtually all cells, but in most cell types it does not occur continuously, but it is induced for a limited time. It is induced by a cell surface receptor activation by the growth factors, integrin ligands, apoptotic cell reminiscent, and some viruses. The ligand activate a signaling pathway that cause a change in actin dynamics and actin dynamics and leads to the formation of cell surface protrusions which are called as ruffles. Collapsing of the ruffle back in the cell leads to the formation of macropinocytosis, right? So this is the first step where you are actually going to have the activation of the cell signaling receptors. Then it is actually going to form the ruffles, and then these ruffles are actually going to rebound back, and that's how it is actually going to form the macropinocytosome. This temporarily increases the bulk fluid uptake of the cell. Micropinosomes non-specifically trap the extracellular fluid along with the other particles and the macromolecules. Macropinocytosis is a degradative pathway where the macropinosomes acidify and then fuse with the late endosome or the endolysosomes, they do not recycle their cargo back to the plasma membrane. So basically, the macropinocytosis is a method by which you can be able to remove some of these fluid from the extracellular media, and that's how you are actually going to it's a degradative pathway where all these molecules are going to be degraded by fusing with the lysosomes or the late endosomes, so that the digestive particles or digestive enzymes from these particles are going to digest. Then we have the exocytosis. So far what we have discussed that how the particles are getting inside, and then how this particle is getting distributed to the different organelles with the help of a process which is called as endocytosis. Now what we discussed the reverse, how a particle which the cell is having and how it is actually going to throw out this process, and this process is called as exocytosis. So exocytosis, the fusion of a transport vesicle to the plasma membrane, is a fundamental process in all the eukaryotic cell. It includes the transport of large molecule out of the cell to the cell exterior, right? So because of the exocytosis, you can even recycle the receptors, you can actually be able to throw the antibody molecules and so on. So transport vesicles from the trans-Golgi network moves towards the plasma membrane, carrying the membrane protein and lipids to provide the new component from the plasma membrane. The vesicles also carry soluble protein that are secreted to the extracellular space. For example, the cell secretes component of the extracellular matrix like the proteoglycans and the glycoprotein through the exocytosis. The exocytosis can include the transport of newly made protein from the ER to the Golgi to the plasma membrane. It can also include the recycling of internalized material from the endosome back to the cell surface. So basically, exocytosis is being used for many purposes. It is being used for secretion. It is being used for putting the receptor back onto the cell surface, and it is also being used for removing the waste material. And in some cases, it is also being used for signaling, right? Because the molecules are going to be produced from this site and it will go to the outside, and then it will be going to induce the signaling into the neighboring molecules. So exocytosis can be constitutive or the regulated. Constitutive exocytosis occurs in all cells via the trans-Golgi network derived vesicles. The pathway occurs continuously. Many soluble proteins like continuously secreted via this pathway, which also deliver the newly made membrane lipids and the proteins to the plasma membrane. So basically, exocytosis is required for delivering the cargo onto the plasma membrane or even outside the cell also. Then what is the mechanism? The empty vesicles actively loaded neurotransmitter onto their lumen using an electrochemical gradient created by the proton pump. Once they are filled, these vesicles are transported to the cell surface for release. Translocation could possibly be diffusion limited or dependent on to the molecular motor. Vesicles are, the exocytosis is triggered by the calcium influx through the voltage gated calcium channels. Presynaptic action potential cause these calcium channels to open. So before releasing their content, the synaptic vesicles dock at the presynaptic membrane at active zone and undergo a priming step to prepare for exocytosis. Secretion is then triggered by the opening of voltage-gated calcium channels in response to the membrane depolarization caused by the action potential in neuron or the receptor potential in the sensory cells. Vesicle fusion and the pore formation are derived by the strong interaction between the conserved proteins such as snares, right, which is called as the N-ethylamine sensitive fusion protein attachment proteins. Snares are the vesicles called V snares, and while those onto the presynaptic membrane are called as the T snares, like the synactin and the SNAP25. So basically, vesicles are actually having the V snares, the target site is having the T snares, and these two are cognate pair to each other. So that's why vesicles will go directly to this particular target site, and that's how they will deliver the cargo. After exocytosis, the vesicles are retrieved through endocytosis. There are at least three known mechanisms of the SV endocytosis. You have the kiss and run endocytosis. You have the fusion pore then closes, allowing the vesicle to be quickly retrieved and reused. And the pathway is fast, requiring 1 to 2 seconds. So basically what happens is that you are actually going to have the vesicles which will go for the exocytosis. They will fuse with the plasma membrane, they will release the content, and then these vesicles are going to be retrieved because they are actually going to filled with the material again, and then will be discontinued, right? So this will continue and it will be keep throwing the neurotransmitter, and these neurotransmitters are then going to be received by the another neuron, and that's how the signal from this neuron will go to the this neuron. So what we have discussed so far, we have discussed about the intracellular trafficking or intracellular transport of the material. And we have discussed about the phagocytosis, we have discussed about the pinocytosis, we have discussed about the detailed mechanism of phagocytosis is relevant in terms of the eliciting the immune responses, and how the post-phagocytosis, you are actually forming the phagosomes, and the phagosomes are fusing with the lysosomes to form the phagolysosomes. And because the lysosome is having the hydrolytic enzymes, it is actually digesting the material what is being engulfed, and then it is going to be presented along with the MSC class molecules so that it can actually be able to trigger immune responses and so on. As far as the pinocytosis is concerned, we have also discussed about the Clathrin-mediated pinocytosis. We have discussed about the Clathrin-independent mediated pinocytosis. And then we also discussed about the Caveolae-dependent process, Caveolae-independent process. And then lastly, we have also discussed about the Macropinocytosis. And subsequent to that, at the end, we have also discussed about the exocytosis, through which the cell is actually throwing the material outside the cell, right? So far we discussed about the internalization, we discussed about the distribution of the molecule within the cell, and then we also discussed about how the cell are going to throw the molecule outside. These outside molecule could be for neurotransmissions, it could be utilized for giving the signal to the neighboring cells, or it could be for recycling the receptor onto the plasma membrane. So with this detailed discussion about the transport of material within the cell or outside the cell, I would like to conclude today's lecture. In our subsequent lecture, we will discuss some more aspects related to this particular course. Thank you.